ISF, and today I'm going to be talking about my research on the seasonal condition and VHSV prevalence in the invasive round goby in the upper St. Lawrence River. So I'm just going to give a quick history on the round gobies and viral hemorrhagic septicemia virus, also discuss my project goals, where and how I collected my data, and then present some preliminary results, discuss my future steps, and then conclude with outcomes and importance of this project. So quickly, the round goby is a benthic fish native to the Mediterranean Black Seas and was introduced into this system by ship ballast water. The first detection of the round goby here was in Lake St. Clair in 1990, and since they have expanded to the massive distribution that you see in the USGS species map here. Round gobies have really become prolific in areas where they are found, and this is due to really a few different factors. First, they've displayed environmental plasticity and have colonized habitats that vary in temperature, salinity, and dissolved oxygen. They present a uniquely extended spawning period from May until August, and during this time they are serially spawning. So you can see that with the presence of both very large, ready-to-spawn eggs and smaller, underdeveloped eggs at a single point in time in this pair of round goby ovaries. They also have a natural, competition-free food source in the co-invasive dracinid mussels, and the overall consequences of these factors are that they've achieved hyperabundance in the system, they have achieved a larger body size compared to their native range, and they've also outcompeted several different species of native fish. So to demonstrate the degree to which they've successfully colonized the St. Lawrence River, I have a brief video displaying their excessive abundance in the habitat. So hopefully you can easily see their massive numbers, as well as the amount of dracinid mussels coating the rocky substrate and littering the benthos. So this video was taken by mounting a GoPro onto the top bar of a 2-meter beam trawl, and this particular transect was at about 15 meters depth near these deep water main river habitats. All right, so the round goby is also one of 28 known species to be vulnerable to viral hemorrhagic septicemia in the virus, and the distribution of this very generalist pathogen is displayed here throughout all 5 of the Great Lakes, as well as the St. Lawrence River, and into some inland freshwater systems, particularly the New York Finger Lakes. Clinical disease symptoms for VHSV include hemorrhaging on the internal and external of the body, pale gills, erratic swimming, and lethargy. In an infected individual, VHSV can present itself in 2 different pathways. An acute disease is detectable by high viral concentrations in a pooled organ sample of liver, kidney, and spleen tissues, and is associated with very apparent clinical symptoms of disease, fast mortality, and is associated with warmer water temperatures from around 9 to 20 degrees Celsius. Acutely infected individuals pose more of a risk of transmission by predation due to those really high tissue concentrations, as well as just decreased predator avoidance ability. So what you're seeing on the side here is really the extreme outward appearance of acute disease in round gobies. This is in contrast to a chronic disease course where the outward appearance of disease is less pronounced in individuals, and the virus is really only able to be isolated from the brain tissues. This is associated with colder water temperatures, typically below 9 degrees Celsius, and these individuals become carriers for the virus, shedding it into the external environment, and therefore they pose more of a risk of cohabitation for transmission. And so in the early studies of the association of round gobies and VHSV, the pooled organ sample was the primary method of detecting the virus, really focusing on that acute disease. And over the course of time, researchers have noticed the importance of testing the brain tissues in parallel to look at these dynamics between acute and chronic disease. So my project is focused on understanding how an individual's condition is related to their vulnerability to the virus. And so in order to do that, I have examined annual patterns of round goby physiology and population age structure, as well as looking at seasonal trends in overall VHSV prevalence, and examining differences in that prevalence between sexes, different seasons, and between sites, also whether the round gobies were caught in nearshore or offshore locations. I'm also trying to look at the differences between these 2 different viral pathways, whether the virus is able to be detected in the pooled organ sample or the brain. And lastly, I'm going to try to use some statistical modeling methods to associate an infected individual's titers to their own physiological variables, as well as to environmental temperature. So my study took place in the upper St. Lawrence River, just outside the northeast corner of Lake Ontario. I have two study sites that are situated about 15 miles apart in the Thousand Islands region. Rose Bay is a more upriver site that presents warmer temperatures, while Cobb Shoal is a farther downriver site and presents colder water temperatures. In each of my sampling bays, I deployed several different sampling methods to collect my round gobies. In each one, I deployed a single trap net, nearshore and offshore minnow traps, and each trawling event consisted of 6 3-minute hauls. I also placed temperature loggers at 1, 5, and 10 meters depth, and each site was also sampled in the nearshore through the 30-foot and 60-foot seine surveys conducted by the Thousand Islands Biological Station. So here I have a breakdown of my sampling scheme throughout the course of 2018, and there were 4 unique sampling clusters. I collected VHSV samples in May, aiming for that 9 to 20 degree temperature range, and also in July and October. And then I collected additional individuals just for physiological data in August. This is a breakdown of the size distribution of gobies that we collected from using so many different gears to collect individuals, and we saw a wide range of sizes. The smallest was about 20 millimeters in size, but the largest individual we collected was 239 millimeters in total length. You can see that the 2 types of trap nets we deployed were the only gear that was effective at capturing the largest demographic of round gobies, while all of the others captured pretty small individuals. And despite deploying 6 different gears, there's still a glaring hole in the size distribution that we were able to catch. We're going to try to supplement that next year by potentially using some angling. We processed in total 545 gobies over the course of the field season, and for each one calculated 3 different condition indices. Gonadosomatic index for reproductive state, hepatosomatic for overall energy stores, and then Fulton's condition factor for just general robustness of an individual. We also examined them all for clinical signs of disease and collected their otoliths to later age by sectioning and staining method. We sterilely dissected a subset of 144 round gobies to test for VHSV, and we collected separate brain and pooled organ tissues and later quantified their viral load by reverse transcriptase quantitative PCR. So just for some preliminary results, the gonadosomatic index showed pretty strong seasonal trends between males and females. With females, a 5% cutoff is used, and males, a 1% cutoff is used to denote a person or an individual is considered reproductively active. So for the females, you can see in May, their mean GSI is well over 5%, and then in July, it's just beneath, with several females still being considered reproductively active. No females presented any gonad tissue in August, and then some rebound and refortification of ovaries had occurred by October. This is in contrast to the males, where they didn't experience any decrease in their mean JSI between May and July, and then again, they exhibited total resorption of their gonad tissues by August, and some refortification in October. For the hepatosomatic index, there were also very strong seasonal trends that differed between the 2 sexes of round gobies. For the males, their mean HSI values maintained relatively similar levels between May and July, increased in August, and then decreased again in October, but overall showed a huge variation in their HSI values compared to the females. The females, on the other hand, showed a strong decrease in their mean HSI values between May and July, suggesting that they're funneling their energetic resources into other systems. And so as we just saw in their GSI values, they are still reproducing during this time and using their energy to be serially producing those eggs. They saw a rebound in HSI in August after spawning was complete, and then those levels maintained the same values, near the same values for October. So these differences in the general trends of GSI and HSI between male and female round gobies really signals a differential energy cost of spawning between male and female round gobies. The overall Fulton's condition factor also showed fairly different trends between male and females. In particular, females were in significantly poor condition in the July time point, again because they were still serially spawning and had used up all of their energy stores. They were just in really horrible condition. They were in intermediate condition in August, and then in relatively similar high condition in May and October. And as we just saw, the components of that condition shifted from gonads to overall energy stores as the year progressed. This is in pretty stark contrast to the males, where their overall levels of condition didn't really change much over the 4 different sampling periods. We found VHSV in both sites, both sexes, and in both nearshore and offshore environments. The highest prevalence that we found was in May round gobies collected from roes, or male round gobies collected from roes in May, and they reached 45% prevalence of the virus. We didn't see any virus detected in individuals collected in October, although there were several samples that were no catch. If you're just looking at the data from roes, it appears that there is a decrease in the nearshore and an increase in the offshore VHSV prevalence as you move from May to July in both sexes. This could be related to seasonal movements of round gobies, as well as the colder deep water habitats allowing the virus to persist for longer. Overall, we found that 11.63% of the gobies tested were positive for VHSV, and of those, only 2 showed the virus in their pooled organ samples, with the remaining 17 only having the virus in the brain, presenting that chronic shedding disease course. So this is a pretty stark contrast to what was found in the earliest studies of VHSV and round gobies, where they were finding high prevalences in the pooled organ samples. So this could maybe signal an evolving relationship between VHSV and gobies over the past 12 years of their association in the St. Lawrence River. Overall, we found significant differences between the 2 different sampling sites and between nearshore and offshore environments, but didn't find significant differences between males and females or between the seasons using Fisher's exact test. However, these were just very quick analyses because we just finished testing last week, where I was pooling all of the relevant individuals into each one of these 2 categories, and wasn't really looking for interactions that appear to exist between these different variables and the months themselves. Future steps for this project are to age all of the collected otoliths and develop length of age regressions, as well as to use statistical modeling methods to associate an infected individual's titers to their physiological variables and environmental temperature, as well as to repeat sampling in the spring of 2019 to compare peak prevalences between 2 different years. The outcomes of this project are an understanding of how the round gobies' changing physiology affects their vulnerability to VHSV, and how in turn that relates to seasonal trends in overall VHSV prevalence. We're also gaining a better understanding of the round gobies' role in transmission to native fish by either those cohabitation or predation mechanisms. And the intersite differences that we detected really signal a need for a spatial scale analysis of VHSV prevalence to identify high-risk areas for future management implications. And it appears that in order to do this, testing male round gobies during that May 9 to 20 degree range would be the most efficient way to achieve this. The importance of this study is to gain a better understanding of the combined roles of VHSV and round gobies on the native community. In particular, the surveys conducted by the Thousand Islands Biological Station have seen a drastic decrease in the catch of young of the year muskellunge that was concurrent with the introduction of VHSV and round gobies into the system. So by examining these relationships of round gobies' physiology, we should hopefully be able to better understand how their characteristics as an invasive species has led to their role in the dynamics of this pathogen in the Great Lakes and St. Lawrence River. There are so many people to thank for this project, particularly the Department of Environmental Conservation for for funding this project and the staff of the Thousand Islands Biological Station for all of their help in collecting this data. And I will take questions at this time.